get this one started. All right, so welcome to, let's see, this is the 14th of 18 talks. So we're going to have 4 more next week, and you're all invited to those. Want to point out for the newcomers to this particular talk that you are all considered friends of the chemistry department and are welcome to come to the chemistry department picnic. You just need to make sure you sign up for the picnic. It's down by the stockroom, and for those of you that don't know what a stockroom is or where it is, it's down by the elevator on the first floor. Okay. So we just need an appropriate head count, and like I said, it'll be next Wednesday in City Park at 4:30. Also want to point out that if you do want to see any of these talks, they are up on YouTube in perpetuity. So if you missed Shelby's talk, don't worry about it. You can watch it. Okay, and point that out. Okay, now it is my distinct pleasure to introduce Emily. And what really makes Emily special was I knew she was coming and knew about her before she even showed up at Lawrence. And this is the first and only student that's ever happened to. Because my next door neighbor is really good friends with her family up north. Okay? And so I was sitting there, next door neighbor's very good friend. He was the one that taught me all about Wisconsin, so I know like how to cheer for the Packers, how to drink beer, all this kind of stuff. So we were sitting there watching Packer game. He's going, Emily's coming up, you gotta watch out for her. And I'm like, Emily? Yeah, Emily Hirn, she's coming up, you gotta watch out for her. Well, it was 2 years of Jack going, watch out for Emily. And Emily and I never quite found each other those first 2 years. She got a different advisor. Jack was outraged that I wasn't the one, but it was a random sort of thing, so I didn't do much about that. But finally, Emily found me, and I became her academic advisor. And it's a great pleasure to introduce her here because it's wonderful watching her grow in confidence in classes over the years. Because she would come in, very first time I met her, she'd worried about this and worried about that. It's like, you're going to be just fine as a biochemistry major. Things are going to work out really well. And then lo and behold, I was right. Okay? And what made it particularly special was then she started taking biochemistry classes. Okay? And I have a lot of Jack stories. And so most people would laugh at a Jack story, but Emily really knows it's true. It's like, oh, Dave, you're bullshitting. No, it really is true. And so it's with great pleasure to introduce Emily for her senior experience talking. It's been a wonderful growth together as well. Very proud, and she's going to be moving on. She's getting married. Okay, fiance is right over here. So that is very special. In addition to going out into the real world and getting a great job, the married part is much more important. So without further ado, I give you.
Like Dr. Dave said, my name is Emily, and today I would like to talk to you guys a little bit about elephants and actually how the herpes virus is affecting their population. So, first off, I want to share some fun facts about elephants because I love elephants. I want you guys to love elephants. I think they're great. I've loved them since I was a little kid and watching Zaboomafoo and all that fun stuff. So, they're the largest land animal, as you can see here, and they can live up to 70 years. They also have really, really long eyelashes, specifically 5 inches long. They also, shockingly, are afraid of bees. I didn't know this until I came across, or until I was starting to do this project and thought it was really funny. Also, they can't jump, so no matter how fast they're running or whatnot, they can't have more than, they always will have one leg on the ground. Also, their trunks. Their trunks are really special because they use them like we use our hands. And when they pick up things, they use them to sense size and whatnot. They also contain 16 different muscles. And they also use them to do things such as hugging. Other fun facts is they actually have very poor eyesight. And shockingly enough, instead of using those big ears that they use their sense of smell as a way to get around. They also use their feet to listen to vibrations from other herds coming. And with those other herds, they all travel in groups. There's a male group and a female group, with the female groups taking care of the calves. They also have very large brains. Specifically, they have a very large hippocampus, which that entitles being able to express compassion, and they are self-aware of themselves. When they look in the mirror, they know it's them and not another elephant. They also greet one another, and what I find really cool is they express grief. They're able to, when they come across a family member's remains, they pick up the bones and they cradle them. And during this time, they're completely silent. You can hear a pin drop. It's so quiet. And they take the time for everyone in the herd to go through them, and then they continue on their way. And also, they prefer one tusk over another, like we prefer to write with one hand over another. So all that set aside, I would like to tell you guys all the environmental roles that they have and how they affect us and all the organisms around them. One thing is that they clear growth in forests. And when they clear this growth, they move trees around and whatnot. That makes it so new growth can come up. And along with this growth, they also have very poor digestion. So while they're eating 400 pounds of food, 50 gallons of water, they also secrete 250 pounds of manure that have non-digested seeds that plant more plants all around. And also, they express this methane gas that could be also affecting global warming. So all these ecosystems that these elephants are in are forests, savannas, deserts, so on and so forth, and more specifically with the different species, Asian and African elephants. The differences between the two is that both sexes, the African elephants, have tusks, while only the Asian elephants, males, have tusks. And also on their trunks, they have 2 points on their trunks that look like that they use as fingers for the African elephants, whereas on the trunks of the Asian elephants, they only have 1 point that they use as a finger. Also, the Asian elephants have 5 front toenails and 4 back, whereas the savanna African elephants only have 4 front toenails and 3 back toenails. More specifically, a lot of people look at the ear's shape and size. For African elephants, they're much larger in shape, and the Asian elephants are much smaller in shape, such as seen here. Here is the African elephant with the larger earlobe and the smaller earlobe here for the Asian elephant. So these wonderful, wonderful creatures, sadly, are facing extinction. One of the facts is with global warming. When the matriarch of the group goes around and she knows what watering hole to go to at specific points every year, and these watering holes she's been taking them to for forever are drying up quicker and quicker, so they have to move more and more. Also, sorry about that. They have poachers that go after their ivory. Specifically with African elephants, they have said that 90% of the population has been killed off in the past century. About 800,000 have been killed in the past 3 decades. And some stats say that 55 elephants are killed a day for these tusks. This is making what is called a genetic bottleneck effect. This genetic bottleneck effect kind of goes something like this. You have this original population that's right here, and then something happens where only a small amount of the population survive. So with these elephants, they have their large original population, then the poaching occurred from humans taking their tusks, and then they have this small amount of population left over. This small amount of population can be affected by anything and be wiped out. Like say this giant tsunami goes all the way over the coast of the continent of Africa and wipes them all out. Like all the African elephants are gone. And this also can happen with viruses. If a virus gets into the elephant population and they can't fight it off, it could kill the species. So the very first case of elephant herpes virus was said to be found in 1988 of a 3-year-old circus elephant in Switzerland. This elephant actually had hemorrhagic symptoms that had blood coming from the eyes and the mouth. I know, kind of gross, really sad for these cute creatures. But they also have symptoms such as shock. So the body can go into shock for any type of infection. They also can have a blue and purple coloring of the tongue. And they also can become lethargic. Their face and neck swell, more so by the eyes. They have a lack of appetite. And like I said before, they have the hemorrhagic symptoms, which is the shedding of the endothelium, so shedding of your blood vessels in your body. These can all lead to pulmonary issues, brain issues, and they can also develop cancer. So with these herpes virus, they're affecting these calves. See, a female elephant goes through a pregnancy of 22 months. Compared to the human 9 months. And they can rarely have more than 4 children in their lifetime. Well, these babies and juveniles that don't have an immune system can die within less than a day of being infected by this herpes virus. And that makes no chance of reproducing, which means no chance for the population to grow. So, knowing all this, we need to know a little bit more about viruses. So generally, viruses are microscopic parasites. They need a host cell to survive. So they infect things such as plants, bacteria, animals, and even, yes, us humans. The different ways they can be transmitted are through the air, an open wound, water, food, and those nasty little insects. So the herpes virus in general, or what I will be referring to it in elephants as EEHV, are an envelope virus. And with envelope viruses, there's an envelope and a capsid, but non-envelope viruses just have this capsid. And the envelope and the capsid protect the DNA. And for the herpes virus, they bring in to the host cell a double-stranded DNA. And most of the time, these herpes virus affect neuron cells in the body. So generally, for a virus life cycle, the virus comes in and attaches to a receptor. The host cell then binds it to the receptor and fuses it in to the cell and kind of goes around it. And as it gets into the host cell, the capsid and the envelope come off, where the DNA goes into the nucleus, where the nucleus makes more of the DNA and also makes mRNA that go to make proteins that make up the structure of the virus itself. And then this DNA and the proteins come together to then assemble and then be released to go and infect more host cells. More specifically, with the replication of a herpes virus, it has a rolling circle DNA replication, which Dr. Dave has a really great way of explaining this, of the toilet paper analogy. So when you use toilet paper, you take some off, then you rip it, and then you fold it, and then you use it. Well, with the rolling circle DNA, it folds the DNA first, then it rips it, and then it uses it. So, all right, we need to know more about this. So what methods are we going to use to measure it? Well, we're going to use PCR, or polymerase chain reaction. The thing that happens here is that PCR denatures the DNA strand that you have into tiny little pieces. And then an annealing step occurs where you have primers that are in here that are specific of what you want to look at. And then there's an extension of when the primers and the DNA that are together, they go along and they make the DNA strands longer. So when you look at these and you use them, you can use them for sequencing or amplifying specific parts of the DNA. So this would be looking at the different types of strains of the virus and to know what it's made up of. And also for qPCR, which knows how many base pairs or how many nucleotides long the DNA is. So to be able to run this PCR, we need to collect some samples. So we need to head over to the elephants and take some tissue. Sadly, some of the tissue that is taken is from dead elephants. These dead elephants contribute heart, lung nodule tissue, spleen, and kidney tissue. The live elephants, mind you, give whole blood samples or trunk washes. They really like to use the blood or the trunk washes because they don't have to harm the elephant, they don't have to poke it with a needle. So with the trunk washes, what they really are is nasal secretions, or for lack of a better word, snot. And contained in the snot is epithelial cells that contain the virus. So what they do is they add a saline solution to the trunk of the elephant. Then they have the elephant lift the trunk up in the air for about 30 seconds. And then they have the elephant pretty much sneeze into this giant freezer bag to then be collected to collect the samples from. So, all right, we do all this, we run the PCR test. So what did they do with all this? Well, they did is they kind of put it in a table and kind of organized it this way. First, they had the virus to know what specific strain they're looking at. So here we are looking at EEHV2 and EEHV3. Then they looked at the strain. Then they looked at the host animal, and here it's adult, male, female, and so on and so forth. Then the location, where was this elephant located? So South Africa. And then they had a pathology of how the tissue was collected. So how they collected the lung nodule. Then they looked at how long the sequence was through the base pairs. And you can see here that not all the sequences were the same. Some were 30,000 base pairs long, while others were about 200 base pairs long. So along with that, when they took the tissue, what they did is they took, right here is a picture of a lung nodule under a microscope. And what they did is they took the little piece of tissue, put it in some type of wax, cut it into thin slices, and then put it on a microscope slide to then be looked at. Here is just what it looks like under a general microscope. Then you got here where you're looking at the virus itself inside the nuclear membranes, and that is shown here in the arrows of each part of the lung nodule. So with all that, we then can look at the evolution of the sequencing from the DNA and tissue. But first, we need to know a little bit about what part of the DNA we need to look at. So we need to look at the base pairs, guanine and cytosine, and adenine and thymine, and which parts are rich throughout the history of the evolution of this. So they found that there's GC-rich strains with a lineage from the proboscis virus, and EEHV4 was one of their examples where they had where they found it with, and they found that it was 206 kilobases long. And then they had an AT-rich section in EEHV1A, EEHV1B, and EEHV5 that had about 180 kilobases long. So with that, they were able to make a phylogenetic tree. And to kind of break this down for you guys, we kind of got alpha here, and alpha is kind of like where the human herpesvirus came from. Then gamma over here is specifically where the human's herpesvirus is. Then other animals are at beta, such as amphibians, reptiles, and so on and so forth. And then down here at delta, we have our elephants and where they came to be in this evolutionary tree. And from this, we can look at to see where the virus might mutate in the future to try to stop it. So with that, we also need to look at how the elephant's immune system is affected. But first, we need to know a little bit about the immune system. So the immune system is made up of an innate and adaptive immune system. For the innate immune system, it's the first line of defense. It is made up of things like macrophages, which is right here. It is a white blood cell, and what it does is it goes through and it kind of eats all the bad stuff. And then you have the adaptive immune system, which has antibodies that come in and fight off the infection, along with B cells that make memory cells, so if you get this infection again, they're able to fight it off. Then T cells have helper cells and killer cells that help wipe out the virus. So the way the immune system normally works is it's about a 2-week period when the virus is in your system where the body really doesn't catch it, but at that 2-week mark, after that, it kind of skyrockets into making a bunch of antibodies to fight it off. Then we have different antibodies that are made up of that increase. So one of them is IgD, which is actually found in milk. Then IgE, which is found in skin. IgM and IgG, which is found in your blood. And IgA, which is found in your intestinal tract and in your respiratory tract. So with all that being said, we need to see how the elephant's immune system is reacting. So what they did is they did a gel filtration system, which is chromatography. And what it is it has different sized beads to break up the different proteins so you can get the specific protein that you're looking for. So as it goes through the column, you collect just the stuff that you want and you test to make sure the stuff you want is there. And you do that by using SDS page and going into a Western blot. So SDS page, or sodium didecyl sulfate polyacrylamide gel electrophoresis, or SDS page, just stick with that, can determine the amount of the major proteins and the size of the proteins. Then you take the SDS page and add it to a membrane for a Western blot. And this Western blot specifically looks at the antibody type that you are looking for. And as they looked at here, when they looked at milk secretion from female elephants, they were able to find IgG response, IgA response, and IgM response in the Western blots that they have here. So these elephants had the herpes virus in them and they were producing these antibodies in their milk. So knowing all this, knowing all the tests that they're trying to figure out how to get through this, how to stop it, they have gone to they have looked at many types of treatments. One of those treatments is, like I said before, the big thing is when your body gets an infection, it goes into shock. So what they do is they give the elephant water, fill it full of IV fluids, and then what they do is they take samples from them to run PCR to see what strain they are looking at to know, to try to see how they can help it better. Then they give the elephant antibiotics and anti-inflammatories to help prevent secondary infection or stop the secondary infection if they already have it. They also have tried oxygen therapy and plasma therapy, and the plasma therapy specifically helps with the intravascular clotting that occurs from the hemorrhagic symptoms that they find in EHV1, more specifically out of the rest of the strains. Then different medications they've tried, yes, they have tried the human medications that, on the elephants. But what they also do is they treat it with antiviral drugs to help it from the cell further damage. And these samples of the drug right here actually have been found to actually kind of stop or slow down the process of the herpes virus from progressing in some of the elephants. When looking at this, they also need to know the way it's transmitted. And sadly, for elephants, it's really hard to prevent the spread because from the trunk secretions, they found that elephants, when they're drinking at the watering hole, playing in the water, splashing water over everybody with their trunks, they can be easily spread throughout the herd because they're all using the same water, same mud, same everything. But they also found in some of the pregnant females that they studied that there was an increased amount of levels in pregnant females, so they can see a mother to calf adaptation for the virus to go in. So, long story short, we need to help these elephants from becoming extinct from this really, really sad state of affairs. And one thing we need is a vaccine, first and foremost. Then we need to find more cost-effective treatments to treat these elephants with more research, and of course, for the vaccines, but also to figure out how to grow this virus in vitro or on a petri dish because they have not found a way to have the virus sustain and grow on a petri dish. And also we need to keep that awareness up for these elephants. Maybe some support on August 12th for World Elephants Day to try to help them and beat this. So I would like to acknowledge some people. I would like to acknowledge my advisor, Dr. Dave. Thank you for everything that you do. I don't know what I would have done here without you. I also would like to thank Professor Sosma for helping me with the presentation. The entire chemistry department, the biology department, and specifically there Cindy and Joanne, who've always been there for me through the ups and downs of this whole ride here at Lawrence. My friends, and of course my family, who have done everything to help me get through this. And here are my citations. Thank you.
All right, questions? Anybody? Okay.
Yes.
So you were talking about a vaccine and that they weren't able to get it to work in tissue culture. Why do you think it is that they can't get it to grow in tissue culture? Because we can grow herpes virus, right? Human herpes virus in there. Why don't you think they can't get the elephant one?
I actually did not run across that when looking, so I don't know. But I would assume because there's so many different strains that's really why, because they can't really pinpoint, because even each strain there's A, B, C, D, and so on and so forth. So I don't think they can really pinpoint to how to grow it due to that.
And I guess the other question is how did it get from, did it start in Africa and go to Asia, or did it go to Asia and go to Africa? How did it, that's a long way.
Well, they see it in zoos as well, but also, and actually some studies have shown that Asian elephants are more affected than the African elephants. They are more affected by this.
Other questions for Emily? Oh, very good.
Are there isolated populations that haven't been affected by it?
That is really hard to study for in the wild. But I do believe they do have some, but of course it's hard to locate. Yeah.
Building on that, have they found that instances are more severe near the human populations and other human sanitation measures that can be taken to prevent the spread, or is it not human spread?
It's not human spread, to my knowledge. And they've actually found that it's the same of infection, like in the wild and in captation. And yeah, they really don't see much of a difference. Yes.
Did using the human vaccine do anything at all? Or did you briefly not?
From what I found, no, not really. It didn't work.
Right, if there are no more questions, let's thank the speaker one more time.